So hi, hello and welcome again, and today's topic is the use of DNA in classification. Fancy title. In other words, I'd like to answer the question, how can DNA help us in grouping organisms together? And here you probably already know this mouse from one of my previous videos, but I do not only have one mouse, I've made 4 of them. Look at this. Okay, it's the one with spots on it. There's a white mouse over here, and oh yeah, this one is also white. And so I've got 4 mice over here, and the question is now, which mice are related to each other? Okay, and just by looking at them, you might say, well, I don't know. This one over here is white, and this one over here is white. So maybe they're related. Maybe they belong together in one group. And the other 2 over here, they have a fur color, yeah, and maybe they belong together. That could be one way of grouping them. But if you remember in my previous video, I already mentioned that is a dangerous thing to do, because we're then only looking at one characteristic, which is fur color. And maybe those 2 mice, maybe they look the same, but maybe they're completely different species. I mean, this could be the possibility, and maybe we don't see it. Yeah. So what scientists are now doing is they're now saying, okay, we want to group the organisms based not on how they look like, but on their DNA, because the DNA, that is more objective. Okay. So there is less possibility for interpretation. If you don't know what DNA is, let me quickly explain this to you. Now, DNA is the genetic code, and every living thing has DNA in the cells. Of course, we humans as well. And this DNA is very long, but it is that all living things have the 4 letters. Well, we don't say letters, but the 4 bases. So for example, there can be A, T, G, or C. And these are the only 4 possibilities. So here it's an A, C, T, A. There's a certain sequence here. And human beings and apple trees and mosquitoes and mice, it doesn't matter. All living things are made of the same 4 bases. But how are we different then? Well, the sequence of those bases is different. It's a little bit like comparing it to a book. There are 26 letters in the alphabet. But there are so many millions and millions of different books that you can write simply by changing the sequence of the letters and by making different words and different sentences. And it's similar in DNA. We only have 4 letters, but by arranging them in a certain sequence, in a certain order, we can get many, many different combinations. And by comparing now the sequence of the DNA, we can now determine how related different organisms are. And what we have to do is we have to now compare the DNA sequence of each mouse here with every other mouse that we have. Yeah? So there are lots of combinations. So we have compare mouse 2 with mouse 4, 2 with 1, 2 with 3, then 4 with 1, 4 with 3, all combinations we have to try out. And when we've done that, and when we've counted the differences in the DNA, then we're able to make a so-called a phylogenetic tree, a relationship tree. And I'm going to show you now how this actually works. Okay, it's not quite difficult, but what I've made here is I've made myself a little table. M stands for mouse. Yeah, so I simply abbreviated that. And what we do is we now compare every mouse, the DNA of every mouse, with every other mouse. And look, what happens when we compare the DNA of mouse 1 with mouse 1 with itself? How many differences are we going to see? Well, obviously 0. It's the same mouse, right? If you compare the sequence with itself, it's 0. So what we're going to do is I'm going to write a 0 in here. Yeah. And of course, when we compare mouse 2 with itself, then there's also it's the same DNA. So there's 0 differences. So yeah, this is the axis of symmetry, as we call it, because everything that's here on the top is the same as everything that's on the bottom. Because when we compare the differences between mouse 1 and mouse 2, then this is the same as comparing mouse 2 with mouse 1, obviously. Yeah. So we don't need the lower half over here. Yeah. So I'm just going to ignore it. I can make an X if I want to. No, I'm just going to leave it empty. We just don't need it. But that's the corner here. That is the thing that we need. And we're going to start off and we're going to compare mouse 2 with mouse 1. So we've already got mouse 1 over here. Here is mouse 2. And what I'm going to do now is I'm going to put them right over each other. And we're now going to compare the similarity of the DNA, and we're going to count the differences now. AA, that's same, CC, same, AA, TT, same. Here's a difference. AG, remember, one difference. Okay, So there is only one difference that I found. Mouse 1 and mouse 2, they have one difference. So I'm going to write it down here. Mouse 2 and mouse 1, they have one difference in DNA. And so it's of course the same down here. Okay. But again, I'm going to ignore the lower half here. Okay. So let's compare now mouse 3 with mouse 1. Okay. Where's mouse 3? Here it is. Okay. Mouse 3 with mouse 1. How many differences are there? Here's GC, one difference, two differences, three differences, four differences, five differences. Mouse 3 and mouse 1 have 5 differences. So I'm going to write 5 in here. And you can already see that mouse 1 and mouse 2 are more closely related because their DNA is more similar. And mouse 3 and mouse 1, they're not so closely related. Maybe one is not even, maybe it is not even a mouse, maybe it's a rat. I don't know. Yeah, small rat. Yeah. So mouse 4 with mouse 1. Where is mouse 4 with mouse 1? I mean, you get the idea. So I can go a little bit faster now. So over here, GC, one difference, two differences, three differences, four differences, five differences. 1, 4 is 5 differences again. Hope I didn't miscount. Again, let's check. 1, 2, 3, 4, 5. Yes, that's fine. So we're all halfway through. Mouse 3 and mouse 2. So 3 and 2 over here. So let's move this again a little bit further down. 3 and 2. By the way, in case you don't know, of course, scientists have computer programs these days that do that for them because they're not just comparing 10 bases, but maybe millions of them. So there's one difference, two differences, three differences, four differences. 1, 2, 3 is 4 differences. And 4, 2, let's compare 4, 2. Okay, 4, 2. One difference, two differences, three differences, four differences. 4, 2 is 4 differences again. And 4, 3, we now have 2. Yeah? 4, 3. How many differences do we have over here? Same. Ah, here, the last one. One difference. 3, 4 is one difference. Okay. So I hope I didn't make any counting mistakes. Yeah. If I did, it doesn't change the principle around. Okay. And now I've got this information. Just by looking at this table, I can already see that mouse 4 and mouse 3 only have one DNA difference. So 3 and 1, they belong to one group. And mouse 2 and mouse 1, 4 and 3, I got this wrong. 4 and 3, they belong to one group, okay? And 2 and 1, they belong to another group. Just by looking at the table, okay? Yeah, because you see there is a bigger difference between 3 and 4 and the others. So, but what I wanted to do now is I wanted to actually show you how you can draw a phylogenetic tree using this information over here to visualize this better, the groups a little bit better. And what I'm going to do now is I start off with those mice that have the closest similarity. So let's say 2 and 1, and then later 4 and 3. So 2 and 1, there is one difference. So what I'm going to do is I'm going to take this table over here. You need a ruler, and between 2 and 1, there is one difference. So you have over here on the x-axis the DNA differences, okay? And over here there is one difference. So what I'm going to do now is I'm going to go all the way over here, and I'm going to draw here, where there's one difference, I'm going to draw here a line, okay? The length of the line is not so important. What I'm going to do is I'm going to go all the way over here into the present. Okay, so I'm going to draw a line like this here as well. Okay, and this is mouse 2 and mouse 1. Mouse 2 and mouse 1. So I'm going to label this here with, and you see I ran out of a little bit of space. So M2 and M1. It does not matter whether you write M1 first and then M2 or the other way around. Okay, it doesn't matter. What is important is that you indicate with this vertical line over here that there is one difference, one DNA difference. And you know what? We're going to do the same thing with mouse 4 and mouse 3, where there's also only one difference. Yeah, so I go again over here. Yeah, so again, one difference here. That's why we're 1. So, and I go like this. And go all the way into the present. And mouse should be the same length. Okay. And mouse 3 and mouse 4. I'm going to write it a little bit here on the side. Mouse 3 and mouse 4, because I kind of need a little bit more space. Okay. So, so far, so good. But now what about the others? Okay, look at this. Mouse 3 and mouse 1 have 5 differences. So mouse 3 and mouse 1, they have 5 differences. So it goes all the way back here to 5. Okay? So let's do that. Okay, mouse 3 and mouse 1 have 5 differences, and 4 and 1, they also have 5 differences. So what I need to do now is I'm going to draw here at 5 differences with a, I don't know, maybe I'm going to use a lighter color. Yeah, no, it doesn't matter. I'm going to use this one over here. I'm going to see that this line will not stay there. Okay, so I'm going to draw over here a very light line going all the way from over here. And I'm going to draw it like this. Okay, like this. Okay, 5 differences. You see over here, 3 and 1 have 5 differences. 3 and 1. So if I want to go from 3 to 1, I have to go all the way back to 5 DNA differences. Yeah. And the same over here, 4 and 1, all the way back here. Okay. But look at this here. Mouse 3 and mouse 2 have 4 differences. So mouse 3 and mouse 2 have 4 differences. So what I'm going to do now is I'm going to draw over here a line at 4. Okay. And, maybe if you're doing this, maybe you would like to use a pencil. Because we're going to erase those lines again. Okay, you see, they separate a little bit, closer here at 4. Now I've got 2 lines, and that's actually not good. Okay, I want to have 1 line. And what do you do in this case? I have a little bit conflicting information over here. Well, the answer is quite simple. You take the average. Okay, so what I need to do now is actually I need to erase those lines, and the average between 4 and 5 is 4.5. So this is actually the place that I need to draw the line, and I'm going to use a different color here. And I'm going to simply erase this here. Okay. Yeah, normally I would do this with a pencil, but the problem is if I use a pencil, then you're not able to see it because it's too light. Yeah. And then I go all the way back into the past, and the line going back doesn't matter how long it is. Okay. But this would then be the evolutionary tree. And this basically shows how those organisms are grouped together and how long ago in the past they separated. Yeah, so you see that mouse 1 and 2, they form a cluster over here, and 3 and 4, they form a cluster, and they separated, all the way back here. And you have to understand that the DNA differences that we have over here, they represent also time. And the further back where organisms separated, the more different their DNA is. Okay, I just wanted to show it to you with this example because over here we had this conflicting information. So my suggestion is that you don't do it like I did, that you use pen first, but that you actually do the math first, that you calculate the average, and then you draw the line there where it is. And then you have a nice evolutionary tree, which kind of, this is known by the way, is known as a cladogram. It's known as a cladogram. And these days what scientists do, I think I already told you, they use computers to make those evolutionary trees. And the cool thing about those cladograms is that we can look at currently existing organisms, and then we can calculate back of how long ago in history when they separated. So what I usually have to do now is I take the mouse 2, I put it over here, that's mouse 2, and mouse 1, that's over here. They form a cluster, they form a group, and 3 and 4, they also form a group down here. Okay, so you see that those two groups are not really very much related. Yeah, so this is basically the little task that I showed you. And of course, I do also have an assignment for you. And the assignment is as follows. And then we're going to summarize everything, okay? The assignment is as follows. It's a little bit simpler because now there are only 3 different animals. Draw a phylogenetic tree to show the relationship between an elephant, a mouse, and a frog. Elephant, mouse, and frog. Which 2 animals show the closest relationship? Well, you don't even, I'm going to give you a hint. Elephant and mouse, they are both mammals. Okay, and a frog is an amphibian. So we can already assume that the elephant and mouse are probably more closely related to each other than to the frog. But we're going to find that out by doing a little comparison matrix again, or at least you can do that. And then you can also try to draw a cladogram over here on, in your notebook, of course. Yeah, or if you want, you can of course also print out something like this and then draw it in here. Okay, so that is a very short introduction into into the use of DNA. But of course, there are a few points that you should remember. Okay, and this can also go, of course, into your notebook. It's very important. Is that the DNA sequence can be used to classify, to group organisms together. This is what I tried to show you. So we try to not look at the external characteristics, at fur color or at size. We try to avoid that, but we try to look at the DNA. And organisms that have a more similar DNA, they're more closely related. Okay, and the classification system should be based on the phylogenetic, the evolutionary relationship, and not on how similar they look. Yeah, it's a little bit like people did that in the past. They made a mistake. They simply looked at the color and, for example, everything that's green is a plant, they thought. Yeah? But in reality, they're also green bacteria, and they're not plants. Yeah? Just because, I always use the car example, just because 2 cars have the same color does not mean that they're the same type of car. Obviously, okay. And it's the same thing also in biology. Just because 2 organisms externally look similar, they might or might not be related. We have to look a level deeper, and that's why we look at the DNA. And that's why DNA is so extremely useful these days, because it allows us to correct also some of the mistakes that scientists made in the past. Because sometimes in the past, scientists misclassified the organisms. They made mistakes because they simply didn't know. They just looked at external characteristics. They didn't know DNA yet. And for this reason, scientists these days are all very often trying to clean up the mistakes of the past. Okay. Yeah. So this is basically a short introduction in how DNA is important in grouping organisms. Yeah. And I would like to encourage you to try this little activity yourself, and of course to put all of the notes and everything also into your notebook. So that's it for today. I wish you a nice day, and see you around next time. Bye bye.